Hey everyone, this is my second video. In this video I will be talking about the career in the field of biotechnology. What are the aspects this field includes? What are the different paths that you can take after BSc? What all knowledge you are going to get? How beneficial it is to get in biotechnology? Scope of biotechnology, job opportunities and much more. All this is answered in this video. So what are you waiting for? Watch, like, subscribe and comment your doubts regarding this field. Now first let's know about what actually biotechnology is. It's a combination of biology plus technology that utilizes biological systems, living organisms or their parts to develop or create different products. For an example, use of yeast, which is a living organism, to produce a desired product. There are main 6 categories related to this field, such as: first, red, which indicates medical and healthcare biotechnology. The next is yellow, which indicates food and nutrition biotechnology. The green, which indicates agricultural biotechnology. The blue, which indicates marine biotechnology. The gray, which indicates classical fermentation and bioprocess technology. The white, which indicates industrial biotechnology. The applications related to the field of biotechnology are correlated to different fields in a tree diagram. There are similar concepts in every field of biotechnology which are being used the most in this pandemic period. For example, the biotechnology and biopharmaceutical companies are the most running companies to produce vaccine for COVID-19. Our only savior of mankind now is science and its application by the biotech industry. This pandemic has really unveiled the true importance of biotechnologists in their industry, but it has also proven itself a big challenge for the medical device sector. On the other hand, this pandemic has opened lots of scope and opportunities for this industry. The tools and technologies that are used are as follows. Now let's talk about the different fields that are included in biotechnology. The first is agricultural biotechnology. It includes the traditional breeding techniques to modify plants and animals or develop microorganisms for specific agricultural use. The crops being developed by Indian sector institutions include bananas, cabbage, cauliflower, eggplant, rapeseed, pigeon peas, sorghum, papayas, cassava, rice and chickpeas. This field delivers significant and tangible benefits for farmers, including increased crop yields and lower input costs. The other way they could be said is they are the genetically engineered products. The second is biosensors. These are the analytical devices that are used to determine the concentration of substances in the body. They generally comprise of a biological recognition element. The most commonly used commercial biosensors are the blood glucose sensors for diabetes management. Now let's learn a technique of cell culture, as it is the most used technique in this field, because all the cure to the diseases are known through the culturing of cells of the infected person in the controlled conditions. Culture preparation vary from single isolated cell to 3-dimensional histotypic cell structures. They are divided into primary, cultures obtained directly from animal tissues, and secondary, permanent cultures of continuously dividing cells. For example, vaccines for deadly diseases like polio, rabies, chicken pox, measles, hepatitis B and COVID-19 are produced using animal cell culture. Recombinant technology broadly refers to the molecular cloning of foreign DNA into bacterial extra-chromosomal DNA elements, that is plasmids, that can be propagated in a bacterial host such as E. coli. Our DNA technology was first applied to protein production in mammalian cells in early 1980s. Chinese hamster ovary, CHO, cells were the first mammalian host for the gene transfer. The proteins generated in CHO cells included beta interferon and tissue-type plasminogen activated TPA. The later became the first FDA-approved recombinant therapeutic protein, Activase, from mammalian cells in 1980s. In biopharmaceuticals, our DNA technology has been effectively used to produce various human proteins and microorganisms, such as insulin and growth hormone, used in the treatment of diseases. Nanobiotechnology. It is the interface of nanotechnology and biotechnology. Nanobiotechnology, a highly topical area of great significance and perspective, deals with control, manipulation, synthesis and biofunctionalization of structures, devices at the nanometer scale.